Welcome to This Explainer, everyone. Okay, today we're diving into a literal medical mystery. We're talking about a microscopic predator that was somehow hiding in plain sight for an entire century, and the wild story of how a pair of rogue scientists completely shattered centuries of established medical dogma. So let's jump right into this, because what we're going to cover today completely changed the way we understand our own bodies.
For the absolute longest time, pretty much everyone, the medical community, society at large, your grandparents, thought they already knew the answer to this question, right? It was just common knowledge. If you had a peptic ulcer, those incredibly painful, life-inhibiting stomach sores, it was simply a case of nerves. The scientific consensus was that intense stress caused your stomach to produce excess acid, and then that acid eroded your stomach lining. Everyone accepted it as absolute fact. But as we're going to see, sometimes facts are completely wrong. And you know what? The science supposedly backed it up. I mean, you kind of have to laugh at the absolute absurdity of how they proved this back then. To establish the stress-ulcer connection, scientists actually took little mice, outfitted them in tiny straitjackets, and lowered them into near-freezing water. And guess what? The stressed-out mice produced more acid. So for decades, the medical establishment just pointed to this bizarre experiment and said, see, stress equals acid and acid equals ulcers. Case closed. But man, this massive misdiagnosis had a real human cost. Because doctors were absolutely convinced stress was the primary cause, patients were often subjected to thoughtless blame. Imagine being in agonizing, unremitting pain, right? And instead of an actual cure, your doctor basically tells you to just chill out. It's all in your head, calm down, you're aggravating your ulcer. Patients were just left to suffer. They were handed useless treatments, told to eat bland, unspiced diets, and drink endless bottles of antacids. It didn't fix the root of the problem at all. Which brings us to part one, a radical experiment, and testing the theory. This is where our heroes enter the picture. Dr. Barry Marshall, a gastroenterology fellow, and Dr. Robin Warren, a pathologist. Now, these guys weren't some world-renowned figures working out of a shiny, major research center. They were working out of a hospital basement in Perth, Australia. They were the ultimate medical rogues who actually dared to challenge the scientific consensus. So look at this wild timeline. In 1982, Dr. Warren spots these weird curved bacteria in stomach biopsies, but they just couldn't get the stuff to grow in the lab. That is, until an incredible stroke of luck. Over the Easter holiday in 1982, a lab tech accidentally leaves the culture plates unattended for several days, instead of the usual 48 hours. And boom, the bacteria actually grew. But here is the crazy part. They couldn't infect standard lab mice. So, in 1984, lacking an animal model and absolutely desperate to prove this bug caused gastritis, Dr. Marshall literally drinks a beaker full of the live bacteria. Yeah, he figured it'd be easier to ask his wife for forgiveness later than permission beforehand. He got violently ill, proved the bacteria was the culprit, and then cured himself with antibiotics. Fast forward to 2005, and that absolute mind-blowing dedication won them the Nobel Prize in Medicine. But, the medical establishment didn't just accept this overnight. Actually, they totally mocked it. Usually a massive medical breakthrough like this is announced in some prestigious, peer-reviewed journal with huge press conferences, right? But the irony here is just too good. The actual cause and cure for peptic ulcers wasn't first celebrated in a medical journal. It was leaked to The Star. Yeah, an American supermarket tabloid famous for running stories about alien babies. Dr. Marshall literally had people walk out on him at international conferences. Changing medical dogma is seriously no joke. All right, part two, anatomy of a predator. Let's officially meet our villain, Helicobacter pylori. This microscopic, incredibly crafty little creature had been hiding in plain sight for a solid 100 years, completely ignored because doctors were absolutely convinced no bacteria could possibly survive in a literal pool of stomach acid. So the crucial question is, how does this bug survive a deadly acidic trap that melts basically every other bacteria? Well, it pulls off an incredible biological heist. Step 1. It produces a super powerful enzyme called urease. This urease breaks down urea in your stomach to create a cloud of ammonia, literally neutralizing the acid right around it. It builds its own little safe house. Step 2. Its spiral shape and powerful little flagella act like a propeller, letting it furiously slither deep into the nooks and crannies of your stomach lining. And step 3, it deploys adhesins. Think of these like microscopic grappling hooks that tightly lock the bacteria onto the cells lining your stomach. And once it's locked on, the invasion really begins. Check out the devastating 1-2 punch of these toxins. First up is CAG-A. The bacteria injects this directly into the human host cell, completely disrupting the cell's physical structure so it literally loses its shape. Then you have the second toxin, VAC-A. This one attaches to the outside of your cell and punches channels right through the membrane. It's drilling holes. And these holes allow vital nutrients like iron and urea to just leak out of your cells to feed the bacteria. It is a complete and total hijacking of your cellular machinery. Moving on to part 3, beating immunity. Because at this point, you might be asking, why doesn't our immune system just wipe this thing out? Like a typical bout of food poisoning, right? Usually, you get sick, your immune system clears the bug in a few days, and you're fine. But with Helicobacter pylori, it is the ultimate biological standoff. It manages this standoff by actively hacking your immune system's communication lines. Let's run through this elite list of bacterial sabotage tactics. First, that VAC-A toxin we just mentioned, it actually inhibits Th1 lymphocytes. Those are the helper cells that normally call in the macrophage cavalry. Next, the bacteria releases enzymes like GGT and arginase that literally stop your immune cells from reproducing, starving them of the vital amino acids they need. Then, it uses a molecule called IDO to block the exact chemical signal that attracts neutrophils, your body's first responders. And the absolute masterstroke, it converts naive immune cells into regulatory TREG cells, which artificially dampen your whole immune response. It literally forces your body into a permanent, lifelong standoff. But look, this standoff is far from harmless. Notice the stark reality presented in this chart here, because this really escalates the stakes. Helicobacter pylori isn't just some nuisance that gives you a tummy ache. It's some major carcinogen. This tiny, microscopic predator is actually responsible for an overwhelming 75% of all gastric cancer deaths worldwide. 75%. Let's just pause on this massive number for a second. 1 million. That is the global impact we are dealing with here. Peptic ulcers and the gastric cancer caused by this single spiral-shaped bacterium kill over 1 million people every single year. For 100 years, this killer was just living right inside us, completely undetected. But thankfully, we arrive at part 4, coexistence and cures, defeating the bug. Because the story definitely doesn't end with us as the victims. Once Dr. Marshall and Dr. Warren actually proved this thing existed, the scientific world finally figured out how to detect and defeat this incredibly resilient stomach dweller. And the way they test for it now is truly ingenious, because we use the bacteria's own survival trick against it. To test for the bug without doing invasive surgery, doctors use this incredible breath test. Step 1. The patient literally swallows a drink containing urea that's labeled with radioactive carbon. Now remember urease, that enzyme the bug uses to neutralize acid by breaking down urea? Well, if the bug is in your stomach, it breaks down this radioactive urea. Step 2. The patient just blows into a balloon. Step 3. The lab checks the balloon for labeled carbon dioxide. If it's there, the bug is there. It's simple, it's non-invasive, and it is so incredibly clever. So once we know you have it, how do we get rid of it? Well, it takes an overwhelming multi-drug arsenal. You can't just pop one pill and be done with it. The CDC recommends a regimen that starts with a proton pump inhibitor. This reduces your stomach acid, which makes the environment much better for antibiotics to actually work. Then, patients are hit with a serious combo of strong antibiotics, stuff like metronidazole or amoxicillin. They even throw in bismuth salts, which you probably know as Pepto-Bismol, to help wipe out the bacteria. This heavy-duty cocktail finally cures the patients who once thought they were just permanently cursed with a bad case of nerves. But I want to leave you with a real curveball today, a provocative twist. Look at this question. Should we always eradicate it? It turns out Helicobacter pylori has been our uninvited silent companion for thousands of years. And because it's so incredibly good at reducing stomach acid, if we completely wipe it out in a person who has absolutely no symptoms, their stomach might actually start overproducing acid. So eradicating this bug in completely asymptomatic people could actually trigger severe acid reflux disease. It leaves us with this wild, mind-bending thought. Are we sometimes better off living in a balanced, steady-state coexistence with the very microbes that have the potential to harm us? Definitely something to think about the next time you get a stomachache. Thanks so much for joining me for this explainer, and remember to always keep questioning the consensus.